morning, everybody, or afternoon, wherever you are, whenever you're listening to this. My name is Ben Jusela, and I am a scientist here at In Vivo Biosystems. And it is my great pleasure to introduce today Chris Lawrence from Boston Children's Hospital. Chris is a fish biologist specializing in the management and husbandry of zebrafish and other small fishes. He runs a very large facility out in Boston. He's worked and managed aquaculture research facilities for over 20 years and currently directs the Aquatic Resources Program at Boston Children's Hospital. He currently serves as a faculty member for the Health and Colony Management of Laboratory Fish course at the Mount Desert Island Biological Laboratory and was a Fulbright Specialist at the Bar-Ilan University Faculty of Medicine in Safed, Israel in 2013. You may also know him as a co-author of The Laboratory Zebrafish, and he's written a number of scientific publications on zebrafish biology and culture. That was a mouthful, but Chris, thank you so much for joining us today.
Thank you for having me, and thanks for that introduction. And I'm happy to be here chit-chatting about zebrafish.
Yeah, when we were like, oh, we're looking for some zebrafish content, and I was like, I know the guy. This format is 17 minutes of science. So I have my trusty Syntago timer here. We're not sponsored. I just got this at a conference, and I'm going to hit go, and we're going to dive right in. Ready to go?
Super.
Perfect. All right. So I know we are talking about zebrafish today, but I would like to talk about the elephant in the room when it comes to zebrafish research, and that is reproducibility. What can you say are our biggest gaps in knowledge and in infrastructure that impede reproducibility?
That's a great question, and it's a question that I think about a lot, right? So, and there's no real short answer, but let me try to give you the 50,000 foot view as I see it. The zebrafish model system is a wonderful model. It has great utility for a number of different scientific disciplines, and it was incredible vision by Streisinger and Charlene Walker and colleagues at the University of Oregon 50 years, 60 years ago when they conceived that potentially zebrafish could be sort of the next genetic model organism. And it's really come to fruition in maybe ways that Streisinger maybe couldn't even have imagined in his wildest dreams. And there are lots of reasons for that. And one of them that's been a real driver for it is how tolerant the fish is to varying environmental conditions, right? So the zebrafish has been in the aquarium trade for probably a century or more, in part because it's really easy to take care of. And it was from the aquarium trade that Streisinger sort of plucked it and brought it into the laboratory in a big way, right? It was probably in the lab before that, but in a big way to build the blocks for it to be a biomedical model. One of the building blocks was that it's just tolerant, it's easy. And that has helped it grow without question, but it's created a problem at the same time. So it's grown incredibly driven by the science and technology and molecular tools and genetic tools, and that's gone way out ahead of everything else. And in the beginning, it was really focused, the use of the zebrafish was focused on developmental biology and embryology. But it's since, as you know very well, that it's expanded to just about, it's the aquatic version of the laboratory mouse. And we're doing all of these incredibly ambitious and complex modeling with this species. And that sort of environmental tolerance, that ease with which we culture it has sort of contribute to the situation where we haven't historically paid a lot of attention to the environment. It's been easy, and it was always just about productivity. But now we have so many things in the environment that we have to think about because the environment, what we're managing in these zebrafish facilities that are now all over the world, those things like diet and pathogens and water chemistry. All of these things are the most elemental piece of any experiment that's being done. And that's where we've made a lot of progress in the last decade, but we still have a lot of work to do in defining these environmental conditions, these extrinsic and to some extent intrinsic factors that impact the science. And obviously I can go on and on about that, but it's all about the environment from my perspective in terms of making it more reproducible.
Yeah, that's really the foundation, right? And if, it doesn't matter if you can technically do something once, right? If it can't be replicated, if it's not consistent from location to location. I mean, how many different background strains and clones of background strains exist around the world at this point? It's a lot, and it's like you said, it's kind of the growth of the system has sort of outstripped the scale up of the infrastructure for it.
Yep. And in that sort of it's one of the beautiful things about the zebrafish model system is that there's no one way to do lots to take care of them. There's no one specific way to maintain them, to make them productive. You can feed them a variety of things. You can keep them under a broad range of different water quality conditions. That in some ways has helped sort of a double-edged sword, right? It's helped the fish, the zebra, when I say fish, I mean the zebrafish, but it's really helped it expand into different laboratories with different infrastructural capacities all over the world very rapidly. That flexibility, it's a beautiful thing. But at the same time, that flexibility is also variability. And that variability, variability is okay as long as we know what it is, right? And so for so long, on the husbandry, aquaculture, environmental side of things, management side of things in the zebrafish world, we haven't really paid attention to that variability. We've just embraced it. We've made it work for us, but we haven't really thought a lot about how these simple things, are impacting ultimately the quality of the science that we're doing. It doesn't mean that we can't solve them. A lot of the stuff is pretty simple. It's a lot. Starts with reporting, right? And just thinking about those things. And that's one of the things that I professionally have been really pushing, particularly in the last 4 or 5 years, is really thinking about how all of these things contribute to the reproducibility and really to the quality of the science that we're doing, because it's my goal to help the zebrafish model realize its great potential. It's like one of my it's what I'm dedicated to. So it's one of the things I'm dedicated to. So.
And you are an excellent resource for the community. I mean, you're very involved with the Zebrafish Husbandry Association. You put on seminars. I mean, when I met you in Boston a couple of years ago, I didn't really know who you were. And it's weird that I didn't know who you were, because you have had your hands all over the place in the model system throughout the years. So I do want to move on. So we got a question from the audience here. I'm going to hold on to that one for a second, because I want to touch on this infrastructure bit next. As you are acutely aware, probably more than many, COVID-19 has been a massive challenge for zebrafish research, from the very small scale facility to the large scale facility. But in particular, it seems to be because of a lack of infrastructure and resources around zebrafish generally. So how did you handle this in your facilities? And following on that, what sort of lasting potentially positive changes would you like to see that have come out of this for the field?
Another tremendous question that we could probably spend a lot more than 17 minutes discussing, but like everybody else, we were really stretched by the pandemic, and have been stretched and continue to be stretched. We've been also fortunate, though, that we've got lots of resources that are allocated to the infrastructure within our institution. such that we've been able to maintain pretty well throughout the shutdown. And I think it highlights one of the strengths of the zebrafish model system that became more apparent to me as I was talking to people all over the world that were sort of going through the same thing that we were early on when the pandemic really hit and everybody was shutting down. And all of these research operations everywhere were just going into hibernation mode. The difference between what was happening in many fish facilities versus mouse and rodent facilities was pretty stark, in the sense that there was a lot in rodent facilities, and this is a generalization, but it definitely was happening. There was a lot of census reduction that was happening, tragically. But it was a necessity that was driven by the fact that that model, that infrastructure requires a certain level of oversight that couldn't be maintained. So there were necessary reductions in the numbers of animals that were being kept. Whereas I will tell you that our census increased. And that was not a unique experience. And part of that is because the zebrafish has different requirements. It's got its own set of complex environmental problems that have to be solved and managed. And these things were deeply thought of, and we worried about them, but we planned for them accordingly. But they have sort of staying power. For instance, they can go for a much longer time without feed. That never happened, but they can without really any meaningful detriment to their welfare. So there are lots of strengths to the zebrafish model that we saw early on that I think were manifested throughout, and it wasn't just us. I think it happened in a lot of places. It also made me think about how much infrastructure. One of the reasons we were able to do it, and I think some of the other places that I knew that did well, if you will, during the pandemic, was that they had the resources allocated so that such the support was there. It maybe was not the same in places that don't devote those same level of infrastructural resources to the programs. And so that's a lesson to be learned as well. The other thing that made me really think about this was how we had to manage, and how many of us have had to manage usage of space, right? So keeping 6-foot distance and trying to keep people safe while rolling back into a research environment was an enormous, and still is an enormous challenge. But it really stressed to me the inappropriate design the design paradigm that we follow now as a community. It's not really we have these 2,000 or 3,000 square foot facilities with thousands of tanks that once you get 2 or 3 people working in one of those rooms, the meaningful work area is saturated. And so you're not really utilizing that well. And so if we had different designs, more modular pod-like designs, for instance, which would dovetail with other things that I think would be important, other levels of control that I think would benefit the model, or certain aspects, certain experimental systems that are being used with the model, I think we would gain a lot of efficiency, but maybe some control over other things that would be beneficial to the field. So it's made me think a lot about facility design. And again, I'm just a representative of the community as a whole. And we have a nice dialogue, active weekly Zoom chats that we have that are free, open to anybody to come to, where we discuss these issues. Really, it's almost like a big lab meeting that started during early COVID, but has continued. But these are themes that are repeated over and over again. And I think my hope is that we take these lessons and we learn from them, and we leverage that information of this incredible stress test that we've been under to make things better. And I think we can, and it feels like we're doing that now. And the other example of it, and I'll let you talk again. This is terrible for you, man. I'm a terrible person for 17 minutes science. Is that this kind of forum, this exchange, it's not in person, but we're talking. We're getting to know one another, we're exchanging ideas. And that is something I think that people complain about, I got another Zoom meeting or a Teams meeting. But reality is that in many ways, we're more connected than we ever were. And I think that I would like to to keep that going, even as we get back towards what is our new normal, hopefully sooner rather than later.
Yeah, I agree, and I think that I think that in addition to being connected and sharing information, it's also an accessibility thing, right? It's, if you're at a small facility, you've got a shelf rack and you're doing it all yourself, like you have access then to a big facility, you have access to the expertise, and you can sort of directly sit there and chat and say, hey, I'm having this problem. What do I do? How do I mitigate this?
Yep.
Same thing with conferences and symposia. Like you don't have to fly somewhere to do this. You don't have to have the budget to do that. And I think that's huge in terms of making this a more accessible system and making the community more, more robust, more diverse.
Yeah, accessibility is really, really an important thing to me professionally as well. Like I want to, some of the work that we've done in the years with the zebrafish system, a lot of it's been done to make the system easier for a broader number of people to be a part of, right? Whether it's, writing or helping to write a textbook, or it's developing a new easier way to grow larval fish. These things are done with the intention of growing the field, but also opening it up to different people, no matter where you are, no matter what level of infrastructure you have, you should be able to leverage it. And so accessibility extends to this medium and the exchange of ideas. And I personally am not, I like the Zoom thing in the right.
When your isn't a contiguous block of meetings, right?
The Zoom meetings I want to go to.
Okay, so with our last minute and a half here, I'm going to roll a question from the audience in with sort of a last question here to close things out. And so the question from the audience is, how do you see zebrafish being part of the model ecosystem? How can it be complementary with other model systems such as cells and or mice? And I want to sort of roll that into my last question of, what do you see as the biggest opportunities for zebrafish in the next 5-10 years? How do they integrate with those systems or complement with them, as opposed to, sort of replacement? I feel like we have this sort of false dichotomy of like, you have to be a zebrafish person, or you have to be a mouse person, or you have to be a cell culture person.
Yeah. I agree with that. I think we've got to get away from that thought and make the zebrafish such that it is just another, as a model, it's another tool in the toolkit of scientists in whatever discipline that you're in. And I think zebrafish apply to lots of different scientific disciplines, right? So it's not just biomedicine. There are other fields in which it has great promise. But I think it should be a tool. It should be one other tool in your toolkit. So you could be less of you could still be a fish person, or you could be a fly person, or a worm person, or a cell person, or a mouse person. But you could also be somebody that utilizes a whole range of these systems. We're off. We're done.
We'll spill over a little bit. We'll let you finish this one out.
Yeah. So I think in order to do that, so I think there is, the expanding utility of the model is really dependent on us making it more reproducible, more broadly accessible, more convenient for people to use, right? So I think along the lines of, particularly as it relates to management, I think getting a standard diet that can be used to standardize nutritional landscape and experiments when needed is important. I think standardizing genetic lines is important, and making them readily available to the community. I think that's a huge issue, and we just had a meeting last week where we talked about that. And so I think and that's what I view as the infrastructure of the model, making all of those pieces of the puzzle more robust, more defined, more accessible, such that we can then really see the zebrafish being sort of fulfilling its destiny or its rightful place in the toolkit for various fields of science.
Yeah, absolutely. I couldn't put it any better, and I have grand hopes for where zebrafish goes in the coming years, especially with people like yourself at the helm helping drive these important points home. So we are at a close here. I want to thank you so much, Chris, for joining us today. I will this has been recorded, so it's gonna go on our website. And I'll also include a link in there to the Friday Zooms.
Please do.
And reach out and access that as well. So
awesome.
Thank you, and I look forward to chatting with you.
Yes, and thanks so much for having me. And I hope we can continue the conversation in lots of different ways in the future.
All righty, sounds good.
Thanks.
Take care, Chris. See you, everybody.
Take care.